Yes, ma'am.
Okay, thank you. Whereas others, only 73 people are here. Any special occasion today?
No.
Rep, are you there? Usually they know, no? Usually they know today 8 o'clock we have lectures.
Yes, miss.
Okay, so 2-3 minutes we will discuss the things that we learned, what are the things we discussed. Anybody? So far what we have discussed? Please recall the things. Not only for me, just recall the things for yourself to understand in which flow we are moving. So we talked about ecosystem, ecology, food chain, food web. Then we talked about pyramids, and then later we talked about different kind of ecosystem: terrestrial, aquatic, and we have discussed under that ecosystem characteristics, classification. Why we are discussing those things? Reason why we are discussing those things? So, I don't like that one-way communication. You should respond for me.
It's related to environmental biology.
It is related to environmental biology. Okay. Why we have to discuss that one? Why we have to discuss?
To know how every ecosystem are differ from each other.
To know how the different ecosystem differentiate from other ecosystem. Okay. Why you have to study the aquatic ecosystem and terrestrial ecosystem?
Different organisms are living in aquatic and the terrestrial, so we can classify them.
Okay, then we have studied the ecological succession. Anybody absent for that class? Ecological succession. I believe that you guys are clever. Your batch is clever batch, and we have gone through your first year, first semester exam papers. Most of the people done well. Soon you will receive your first semester exam results, right? And I need more interactions with you. So please respond for the questions and the discussion. No ideas? No ideas? Okay. Today we will discuss, yeah.
Ecological succession means species change over the time.
Okay. Last class I asked one question. Differentiate the ecological succession and evolution. Isn't it?
Yes, ma'am.
Yes, ma'am.
So can anybody tell me the differentiation? Evolution and succession. No idea? Evolution and succession. Did I ask that question? Didn't I ask?
You have asked me.
Yeah. Then tell me the answer. What's the difference between evolution and succession? Somewhat it's look like related. Somewhat it's look like same, but there are different. Can you tell that? Okay, simply I will say in two keywords. But whenever you face the exam, question in the exam, you have to explain briefly. Right? The succession is related with species differentiation, that modification in the species, how they adapted to the environment or ecosystem. Evolution is related with gene, that is gene modification, right? So it is gene modification adapted to the ecosystem. That's a major difference. Species modification and gene modification is evolution, right? So we will move into the today's topic, bioindicator species and their uses in monitoring in the ecosystem, monitoring of the ecosystem. What do you understand from the title, the today's topic? The thing, one person sent message. When you are sending messages while discussing, I have to open it and I have to read it. If you unmute and talk with me, we can understand directly, right? Yeah, evolution is slowly adapt to the environmental stress over time by species. Succession is modification. Yeah, both are modification. Mainly, the evolution is based on the gene modification, right? So succession is in the species adaptation than the modification. If the species getting modification, it's definitely related with gene, right? So evolution is directly gene modification. Succession is the species adaptations, right? Okay. So what do you understand by bioindicator? Bioindicators. Any ideas? Okay, it's indicate the present condition of environment. Yeah, good. Any other response?
The bioindicators.
Yeah, bioindicators.
Living organisms such as plant,
Yeah, good.
animals.
Bioindicators. The word bio, that means it's talking about the organisms. It may be plants, it may be the microorganism, macroorganism. Bio means they are going to indicate something as they are organisms, right? So what are the bio indicators are there? The classification of, so today we are going to discuss what is bio indicators, then classification, then their uses. So these three things we are going to discuss, right? So bioindicators, species. So bioindicators are the organism that indicate or monitor the health of the environment. So they may be used for indicate or the monitor. Through the indication, we are going for the monitoring of the ecosystem. So what we are indicating, what we are monitoring is the health, that mean health of the current situations and health of the ecosystem. Right. So those indicate, that bioindicators indicate the presence of the pollutant and also attempt to provide additional information about the amount of intensity of the exposure. Right. So from this, the new word, so word pollutant. So pollutant is the substance which is creating pollution. Right. So in next class we will discuss the pollution. So next class topic is pollution. Right. So this may help in the pollution control system. Right. So the basic thing is bioindicators species indicate the presence, so they may indicate the presence of pollutants. Why they are indicating the presence of pollutant? Through the intensity of the pollution and how they expose in the ecosystem, right? And according to the bioindicator characteristics, how they are present in the ecosystem. So if it is dense amount of bioindicators or low amount of bioindicators, the characteristics of bioindicators, how they are behaving. So their behaviors also indicating some kind of information for us from the ecosystem, right? We will move into the classification of bioindicators. So we will classify the bioindicators based on the aim of indicators, based on the applications, based on the IUBS, right? So these are the classification of bioindicators, right? So first one, based on the aim of indicators, right? So aim of indicators. So if it can be compliance indicators, diagnostic indicators, early warning indicators, right? So compliance indicators they focus, so where we are using the aim of the usages of indicators, right? So compliance indicators they focus on the issues such as the sustainability of population. So when we are discussing about a particular population, maybe plant population, animal population, so any other microorganism population in a particular area. So in that time, when we focusing, sometimes we may get information that this species particularly, we can say. So for example, in Trincomalee, right? So we know Trincomalee is famous for deers, right? So you can observe the deers near to the Koneshwara temple and beach, Trincomalee beach. But nowadays the species density of deers are getting down, isn't it? So it is related with the issues of the sustainability of the bee population in the particular area. We can say near to the beach area, near to the temple area. Why it's getting? So 1 second. Right. So if we want to study the particular population sustainability, so we can go for with the compliance indicators. So there are some kind of indicator organism will indicate why this particular population is getting down, why the particular population is getting low density in a particular area, what happened to the ecosystem. So we can analyze those things through the compliance indicators. And second one, diagnostic indicators. So diagnostic, the word diagnostic will indicate the meaning, diagnostic indicator. So they mainly used for to measure on individual or sub-organisms, right? So if we want to diagnose one individual, so what is going on within the particular species, what is going on within the particular populations, are they going in a sustainability manner, or are they facing any difficulties to survive in particular ecosystem? So if we want to diagnose those things, we will use the diagnostic indicators, right? And early warning indicators. So under the aim of indicators, the aim of usage of indicators, third one we can say early warning indicators. So early warning, so they will give some kind of warning response which will help us to get aware on the what is going to happen in the ecosystem. So mainly they focus on the rapid and sensitive response. So see, rapid and sensitive response, that is giving the early warning. If they are slow or if they are not sensitive, so they can't used as indicators. That means if they are getting any disturbance in the ecosystem, if they are getting any damages, or if they are feel anything, any changes in ecosystem, they suddenly respond to that particular changes. So they are rapid and they giving a rapid and sensitive response to the environmental changes. So when the environment is getting changed, they rapidly give the response. So these are the three indicators based on the aim of usage. So compliance, diagnostic, early warning indicators. Right? So second one, based on the application of indicators, right? How we are going to apply the indicators, right? So we will discuss that one. So environmental indicators, ecological indicators, biodiversity indicators, right? See the words in telling the meaning of the indicators. The environmental indicators. Right, so environmental indicators. So these are the species or group of species responding predictably to environmental disturbance, changes. Right, so environmental indicators mean if there are any changes happen in the environment, they suddenly give the response. So maybe it may be predictable, right? So predictable to environmental disturbance. So for example, we can say sentinels, detectors, exploiters, accumulators, bioassay organism. There are several researches on the bioassay organisms, right? So these are environmental indicators. Can you tell any examples that you have observed in the ecosystem as the environmental indicators? Environmental indicators. Any environmental indicators? Don't you observe any environmental indicators from your childhood? You people are listening to me or not? Yeah,
Yes.
tell me some of the environmental indicators you observe.
Butterflies.
Butterfly. Okay, so in which situation you will observe butterfly? So normally butterfly is an organism, right? It is an organism which is surviving in the ecosystem. How you are saying that is maybe a environmental indicators? Reason.
In the spring season,
Okay, in the spring season.
flowering season, full of environment.
So you are saying in the flowering season, butterflies will get high population, or because of the high population, you are saying that is spring season.
When the atmosphere is popular,
Atmosphere.
less polluted,
Yeah, less pollutant. So we can say if the amount of the particular organism, for example, butterfly amount is high, we can say the characteristics of the ecosystem, the environment, isn't it? If higher amount of butterfly is there, we can say that area should be less polluted, right? So that area facing low pollution under some circumstance. So and some kind of thing we can say it is a flowering season. That's why the butterflies count, so butterfly amount is high, right? So according to the some kind of organisms behaviors, population densities and changes and behaviors of themselves, we can say the characteristics of environment. That is meaning environmental indicators. Clear? You clear with the meaning of environmental indicators?
Yes, ma'am.
Okay. Next one is ecological indicators. So see the difference between environmental indicators and ecological indicators. So this species that is known to be sensitive to pollution, so habitat fragmentations or other stresses. So the response to the indicator is representative for the community. Right? So what's the major difference between environmental indicators and ecological indicators? Both the same or it has some difference?
Environmental indicators that is not sensitive to pollution. Ecological indicators that is sensitive to pollution.
Yeah, great. So mainly environmental indicators, somewhat it is indicating it is used as a detector or bioassay. However, the ecological indicators mainly focus for the pollution. So they are mainly sensitive to the pollution. So we can use those indicators for the ecological indicators as a pollution indicators, the pollution analysis, pollution monitoring. Right? So normally these ecological indicators working for a community. Right? So that's the major difference between environmental and ecological indicator. However, those are little same. That's the changes happen in the ecosystem. Right? Third one is biodiversity indicators. Right? So biodiversity indicators. So those are indicators for species richness of the community. So biodiversity, the meaning of biodiversity, you know, the diversity of the species. So when the indicators is exposing the species richness of a particular community, those indicators known as biodiversity indicators. That means if species is letting us to know about some species diversity, so some species richness. So from the species richness, if we are evaluating the biodiversity, those species are known as biodiversity indicators. However, the definition has been broadened to measurable parameters of biodiversity, including species richness, endemism, genetic parameter, population specific parameter, and landscape parameter. So it can be, so in these categorization, we can say we can broadly discuss about this biodiversity. So if it is talking about the species richness, so in this way we can talk about the biodiversity. We can talk about the biodiversity in the manner of genetics. So through the genetics also we can discuss about the biodiversity. So if the indicators is mainly focusing on the genetic parameters, so we can say that is in the way of genetic parameter through the biodiversity evaluation or monitoring. So in these ways we can categorize through the application of indicators. Right. Third one, based on IUBS. You know what is IUBS? IUBS. Any idea? It is International Union of Biological Science. Right? So based on their categorization, they classify the biological indicators: microbial system, plant system, animal system, cell biology, and genetic system. So we will see one by one. Microbial system. right? So in the microbial system, so these are diverse group of organisms found in a large quantity. So those are diverse group of organisms which are present as a large quantities. So the characteristic of that microbial system is easier to detect and sampling. So we can use it easily to detect and sampling, right? And sampling means, so if we want to take the evidence, so if we want to analyze a particular ecosystem, we have to take the samples. Samples mean we are taking some evidence from, we are collecting some specimen. So those collection of specimen we are calling sampling, right? So the presence of some microorganism is well correlated with particular type of pollution, and it serves as standard indicator of pollution, right? Mainly, so microbial system we can use for evaluation and monitoring of the pollution system. So mainly, if a particular microbial system is growing, or if we found a microbial system in a land area, so we can say this indicating some kind of pollution. So those will survive in the pollution area. We will discuss further, right? So bioluminescent bacteria as indicator, right? Have you seen this picture? Have you seen this kind of organisms or this kind of culture plates somewhere? See, the bioluminescent, that means it can create luminescence. So it can create some energy which is looking like that light energy exposure, right? So these are used to test water for environmental toxins. So bioluminescent bacteria. So for example, if we guess, or if we suspect if any water body getting some toxic, right? Not if that area is polluted with some toxin, we can go for the analysis with the bioluminescent bacteria. We can take the sample of the particular water ecosystem, and we will go for the culturing methods of the particular water sample. So you have studied the culturing methods in laboratory techniques. Am I right?
Yes, ma'am.
Yeah. Can you tell the culturing methods? What are the culturing methods you have studied? Because we have to correlate the things what you studied. How we are going to use that culturing method, culture plates you have studied in laboratory techniques. So we are going to discuss how we are going to use those culturing methods here. How? See, first one is different culture method than others. See, this plate and these plates. Those are same culturing method?
No, ma'am.
No. Then tell me what are the culturing methods you have discussed in laboratory technique? Plate method, good.
Street plate.
Four plate method. Yeah, streak plate method. So first one is in which category?
Street plate.
Yeah, streak plate. Then second, others, how you can say?
Four plate.
Yeah, it's look like four plate method, right? So we can go with those culturing methods, right? So different organism having different culturing methods, and can you use all media for all microorganism growth? So what is the basic media used for the bacterial growth? Nutrient agar. Any other answer?
PD point.
Okay, we can grow that in potato dusters agar as well, right? So from this you have to know. So when we are choosing a particular organism, we have to know that media, the culturing methods and condition, incubation period, incubation method, those things, right? So after collecting the sample from the water body, that polluted water body, you will receive the sample, and our guess is what. From this sample, we are going to analyze whether there are bioluminescent bacteria is present. If the bioluminescent bacteria is present, we can confirm that is polluted with some toxin, right? So the meaning is the bioluminescent bacteria can grow in particular toxins, right? So through this process, we can evaluate the particular water body whether it is affected with toxin or not. So if there are toxin present in the water, the cellular metabolism of bacteria is inhibited or disturbed, right? So as we can say in some other organisms can grow with the survival of toxin, but some other organism will not grow, so it can be inhibited. That means metabolism of bacteria can be inhibited or disturbed. So this affect the quality and amount of light emitted by the luminescent bacteria. So here the concept is, so normally we can have a control plate. Control plate means we will grow that organism in a distilled water. That means there is no any toxin, right? So other one is our particular experiment. So in that experiment, there may be distilled water, there may be polluted water. So we after a period of time, so we will allow this for a period of time, and we will evaluate that one. The intensity of light emission. That means bioluminescence means intensity of through the intensity of light emission. We will compare this one. The control one will have the high intensity. That polluted water sample will have the low intensity. So then we can conclude that one. So this particular water sample is affected with the pollutants which is having some toxins, right? So it is very quick method. It can take just 30 minutes to complete. That means within 30 minutes, these bioluminescent bacteria will populate and make as the cultures, right? So some bacteria will take time. Maybe it can be take one day, 2 day, 3 day intervals. So you have to frequently analyze the culture phase. So this is related with the bioluminescent bacteria from the polluted water. So if the intensity of light emission is reduced, we can say that is polluted water with the toxin, right? Next one is plant system. So the presence or absence of certain plant or other vegetative life in ecosystem. So we can say presence, absence. Likewise, here also, here we can say the toxin maybe suppress and inhibit the growth of the metabolism of bacteria. In some other researchers, they mention some bacteria can grow in a particular toxin circumstance. So if those bacteria is growing in the experiment without growing in the control, if it is growing in the experiment, we can conclude that one that is having particular toxin or particular pollutant. Likewise, in the plant system, whether it is presence or absence. So some kind of plants may be present in an ecosystem with the pollution or non-pollution condition. Likewise, the absence of the plant system in a healthy ecosystem otherwise in a polluted ecosystem. So it may be in two ways. We can discuss it in two ways. So they can be formed both higher and lower classes of plant base. Right? So lower plants means the susceptibility of the resistance towards a substance in the environment varies with the species. So the resistance, resistance, susceptibility, and moderate susceptibility, moderate resistance, those things. So resistance means they resist. Susceptibility means they accept the things and getting weak. Right? So the meaning, if we get in COVID, we are susceptible for COVID. That means we are getting the organism inside our body and we are affecting with that. If we are not getting COVID, that means we are resistant to the COVID. So like that, low plants, the susceptibility of resistance towards the substance in the environment varies with the species. That means susceptibility and resistance is not same for all species. They are different, right? So for example, lichens. So another one is higher plants. So higher plants, the sensitive species are employed to detect and monitor specific air pollutants. So we can say there are studies on higher plants are more specified on its ability to indicate the heavy metals population in water. Right. So last year I have done one research by using the aquatic plants. So do you know we can measure the pollutant or pollution intensity of water body through the aquatic plants? Can we? Have you observed in aquatic water body, we can say ponds. So if we observe some kind of plants, how can we evaluate the ecosystem? Tell me some aquatic plants names.
Hydrilla.
Hydrilla, good. Sorry, it's not clear. Personally, okay. Iconia, great. Lotus, yes.
Salvinia.
Salvinia, okay. Good. So we can categorize this according to the species availability and species density. We can say a particular pond's characteristic. For example, have you observed a greenish ponds?
Yes.
So what does it mean?
Eutrophication.
Yeah, eutrophication. So we will discuss how this eutrophication is happen. So from the eutrophication, what is your look like? What is you observing as a green color? The floating plants or floating organism on the water body. What's that?
Algal bloom.
Yeah, great. Algae. So the meaning of that is, so algae, they are in that particular circumstance, the algae is the biological indicator. That means if the water is getting eutrophication, that means the nitrogen and nitrates and phosphates, that density is increases. So for example, so from the industrial discharges, so if the pond area getting more amount of phosphates and nitrates, what will happen? Nutrient contents of nutrient intensity or nutrients concentration will increase in particular pond area. So then the algae will bloom. The algal blooming is creating the eutrophication, right? So that's the meaning of that indication by the algae of the eutrophications, right? So lower plants. The lower plants, for example, we can say lichens, alga and fungi living symbiotically. So they have to live together to survive. So these live in extreme conditions, but they hate pollutions. So they can survive these algae, fungi, so the lichens, they can survive in the extreme condition. That means in extreme level of pollution also they can survive. But it doesn't mean that they are liking pollution. They hate pollution, but they have the high level of resistance characters among themselves. See here, this pond. Can you see the algal blooming? This is the result of eutrophication, right? See, from this indicator lichen, see, these are in this arrow. This area is leafy lichens, brown. See, crustose lichens, bushy lichens. So, bushy lichens mean if the moisture content is high, you can observe the bushy lichens. If the moisture content is low, you can observe the leafy lichen and crustose lichens. See, so these are the differentiation of their indicator lichens. So the cleaner the air, the bigger and more elaborate the lichens, right? And we will discuss mosses and lichens, how they are used as the biological indicators. So we can say the indicators of environmental pollution due to their capacity to accumulate and store heavy metals and store heavy metals and other toxins. So it's typed in 2 times, right? So that means the lichens and mosses are, it can be accumulated in a particular area. So according to the capacity and according to their density. So we can evaluate the ecosystem or environmental pollution, and they may be stored the heavy metals and toxins, right? So the typical example of biological indicators of air pollutions are lichen, right? Their the major advantage is the response repeatability is so in various habitats, right? See here the color change. See, according to this, we can say that area is getting pollution or that area getting damaged. See, it's in a normal condition. See, this lichen in a normal condition. This is in the pollution condition. See here, this character, this color and the density is in normal condition. See this one, it is in pollution or polluted area. Can you differentiate? Likewise, we can say it indicates the ecosystem, health of the ecosystem. Changes in diatom community. So the decrease in plankton, algae and aquatic hydrophytes indicated increased water acidity. Right? If you observe the changes in diatom community or decreases in plankton, algae or aquatic hydrophytes, that organism which is surviving in the aquatic ecosystem. So if you see the reduction of population of these kind of organisms, we can say the water body is getting acidity. Right? So increased acidity will cause this problem. And specific changes in aquatic flora can indicate the pH of the fresh water correctly. See, we can say that aquatic flora changes, maybe we can say the population changes or community changes or their behavioral changes can say the pH of water changing or pH of water of the fresh water. Right. Next one we will discuss the higher plants. Right. So the chloric flakes of pines. This one. Chloric flakes of pine needles are good examples, which is indicating the ozone damage. Have you observed this one? Pine trees, have you observed?
Yes, ma'am.
Have you observed this kind of symptoms? See, In coastal area, we can find the pine trees, right? So if we are observing the chlorotic flakes, see, these are the chlorotic flakes. So chlorotic flakes of pine needles, it's a good example for the ozone damage. So it's because of the ozone damage. And we can say the collapse, glazing and bronzing of leaf cells are products of damage by PAN, PAN. So we will discuss this PAN in pollution discussion, and that means peroxyacetyl nitrate. So these bronzing and glazing of leaf cells can be the result of the accumulation of the PAN. So it's creating from the PAN damage, right? And the grasses, cyanidin dactylin, was evaluated as a biological indicators of heavy metals such as lead, copper, cadmium, manganese, zinc, chromium, and nickel. So I have started one talk about my research. So I've done a research through the aquatic plants, right? So the two things, aquatic plants are accumulating, that means absorbing the heavy metals, some kind of heavy metals from the water bodies, and when they are getting the accumulation of the heavy metals from the water body are getting purified. Understood? So there are some researches. So if you are interested in this kind of work, go and search the aquatic plants usage in the purifying water bodies. So that meaning the basic theory plant, for example, hydrilla. So we can say that hydrilla roots, they are absorbing some kind of heavy metals, right? So my idea was, if they are growing the aquatic plants in a particular plants, if they are absorbing the heavy metals, we can clean the water body by using the aquatic plant, isn't it? Any idea while I'm saying this one? No, any idea? Understood what I told?
Yes, ma'am.
Yeah, what's that? So don't think all the aquatic plants will accumulate the heavy metal. Some kind of aquatic plant has the ability to accumulate or absorb the heavy metal. So we have to evaluate the aquatic plants, which are the plants or has this the ability. Right, so I used 5 plants, right? So 5 aquatic plants, and how you can ensure that those are accumulating the heavy metals? How you can ensure that one? Think
In
how
the water body filtration.
water body nutrition. Okay, first you have to analyze. So in the water board, water boards of every province has water board. So water board has some other experiments. So evaluation of heavy metals, nutrients, some other things. Right? They have some rates. So in 2000, I done this research in 2020. Right, on that time, to evaluate lead 2500 for one sample. If we have, so normally for experiment we have 3 replicates. So if we go for 3 replicates for one plant, so for 5 plant, 15 replicates. Am I right? 15 replicates. So 15 replicate into 2500, like that we have to do that experiment, right? So you can go and give the samples. I'm doing this research, and they will give they will go with the analysis, the evaluation, and they will give the result of the analysis. They will go for the analysis and they check that whether if I am asking to check them to evaluate the copper density, lead density, then they will analyze this and they will give the report. From that report, we can analyze if which plant accumulated more amount of heavy metals. Then we can go with that particular plant to treat the ecosystem, that pond area. So it is not promoting the aquatic plants in the ecosystem. Some other aquatic plants creating pollutions. Right? So that meaning is some aquatic plants have the ability to absorb or accumulate the heavy metal. Then we can use those plants for the monitoring and cleaning or purifying the ecosystem. So that's the basic concept of that research. Right? You also can think some kind of ideas. So now don't think we are in first year, second semester, we can't do researches. You also can do researches. See, biological indicators. So you can go for survey. So now you are in home. So you can select your area. So if you are in Trincomalee or if you are in particular area, you can go and check the plants. Ponds presence or ponds situated in your area. So you have defined your experiment area. Then if there are 10 ponds, right, you can go to the ponds, you can check the health condition of the pond area, the plants present in the pond area, density. So you can take a survey. From that survey, you can go for the analysis, monitoring what is happening in this area, and you can ask from your site agriculture department or any other ministerial department to take the past histories of the ponds, right? Then you can analyze the things and you can conclude the conditions or the changes happen in the particular area. Not only for the ponds, if you want to go with the forest also, you can go with the forest studies. Right, the availability of plants, how they are surviving, the changes happen over a period. So those also some kind of researches. If you use your free time with these things, you can write a paper, research paper. So if you write a research paper and publish in a conference, then you will get more points. The thing is, who are those focusing to become lecturer or become scientist, become a researchers? If you develop from now, it will help you in future, right? So it's my advice, right? So next one is abundance of iconia. So the abundance of iconia will indicate the sewage and heavy metal pollution of water, right? And equisetum species. So the presence of gold in the soil. So these species indicate the presence of gold in soil. And we can search the species of plants and you can grow. If it is grow, that soil has the gold availability, right? Annual weeds and short-lived perennial like amaranthus, xenophodium and polygonum. These annual weeds and short-living perennials, they are indicating overgrazing. Right, they are indicators of overgrazing. Next one, we will move to the animal system. So, animal system, an increase or decrease in animal population, it may indicate the damage to the ecosystem caused by the pollution. So, all the things are indicating the damage to the ecosystem. Sometimes it may be the environmental changes. Sometimes it may be anthropogenic changes through the pollution. So pollution not only through the man-made, so through the anthropogenic activity. Pollution can be created from the natural changes also, right? So monitoring the size and number of certain species. So other mechanism of animal monitoring. So it can be talked about the concentration of toxin in animal tissues or monitoring the rate at which deformities arise in animal populations, right? So earthworm. So normally we know earthworm is a farmer friend, isn't it? So earthworm density and the biomass determine the acute toxicity. So normally we can do the experiment, health soil with the polluted soil, right? So for if you want to analyze the pollution density or pollution evaluation of these particular soil, so we will take the soil, healthy soil and polluted soil, and you can leave 10 earthworms in health soil, 10 earthworms. So when you are leaving 10, those 10 should be equal in size, equal in length, equal ages. That means 10 days old hatchlings or 30 days old earthworm. So like that, it should be same, right? Then only we can compare and contrast the characteristic. If you are putting here old, so sometimes if you are putting young juveniles or young hatchlings in the good health soil, and if you are placing the old earthworms in the polluted soil, can we say because of the pollution it got died? Can we say? Are you listening to me?
Yes, ma'am.
What I told? How we are going to measure the polluted soil? These are the basic research experiment rules, right? So if the then we can say if the density and biomass is reduced, then it has some toxin in the soil. So the determination of toxicity by the earthworm density and biomass. And frogs. So frogs are the environmental stress and health of biosphere as a whole. So we can say if their behaviors, sounding, according to the sounding also we can say it is in stress, it is in the reproduction mood, or it is the rainy season is coming. So frogs will create some sounds. So according to the sound also, the environmental condition will be indicated. And some kind of fish. So some kind of fish will indicate the water pollution. And macroinvertebrates, so very sensitive to pollutions, excess nutrients, increased turbidity, chlorine, and etc. So when the behavioral changes, the intensity or density or biomass is changing, so we can say there are that amount of nutrients, whether it's increasing or decreasing, turbidity, chlorine content of a particular water body, right? So invertebrate as bioindicators. So far we have talked about the other organisms. So how we can use the invertebrate as bioindicators? See here, first image. This is the maggots. So see, rat-tailed maggot, which is lived in sewage polluted water. So sewage tanks. So some other area you can observe this one. So if you observe this rat-tailed maggot in the RO water plant from the hostel taps, so we can say that is contaminated with the sewage tank or sewage water. So it can be evaluated through the rat-tailed maggot. And flatworms. So flatworms also live slightly in polluted water. That means they can survive in that water. So that if they are surviving, or if we found a particular organism in the area, we can say definitely it's a polluted water. And freshwater streams, they can live only in fresh water, right? So if you want to measure the fresh water, so whether some water samples are given for you to measure whether which one is fresh water, then you can place the shrimps in each and every water samples, and if the shrimps is survived in the water, that water will be fresh, others are not fresh water. Understood? Like that you can do the experiment, right? Last one, the cell biology genetic system. So cellular and subcellular components, so chromosomes and adapted to specific environmental conditions form an excellent parameters for the indicator. So many animals shows behavioral responses following the detection of environmental changes in the functioning endocrine, nervous, in the nervous systems and muscular, cardiovascular and excretory system. So such changes may be investigated at the morphological, biological, physiological level, and also it can indicate the presence of toxic substances. Right? So criteria for selecting bioindicators. So far we have discussed about the biological indicators, the usage, applications, the aim, right? So how we are going to select the indicator to a particular area. So for example, you are getting some complaints. So as a researcher, you are getting some complaints. This pond area having this kind of plants in this density for a particular period. Then as a researcher, what's your responsibility is you have to monitor and evaluate that particular area. If you want to go with the usage of bioindicators, you have to select a particular indicator. You can't go with all the indicators, right? You can't go with amaranthus, using amaranthus to check the water quality. Can you check the amaranthus used to check the water quality? Amaranthus. I think time exceeded, no? Okay. So quickly we can wind up. So according to the sensitivity, specificity, broad applicability, and representativeness and cost, these are the things you have to consider. And uses of bioindicators. So forecasting of disaster. So for the disaster analysis, prevention of pollution, exploration, conservation of natural, sustainable development with minimum destruction of the biosphere. So, and also predicting the impact of anthropogenic activities, particularly pollutants, predicting the environmental change in a timely manner. Okay, so these are all about the bioindicators. Sorry for that. I got time 4 minutes excess. Okay, so we will discuss in next class further. I will wind up today's class. Thank you.